Have you ever wondered how a tiny change in just one cell can lead to a deadly disease like cancer? Today, we're going to explore how disruptions in cellular biology can cause appendix cancer.
Let's start with the building blocks of life, cells. Like a factory, each organelle performs a specialized role to keep everything running smoothly. The nucleus is the control center of housing DNA that directs all cell functions. Then the plasma reticulum, ER, and ribosomes build proteins, some of which regulate cell division. The Golgi apparatus packages these proteins for transport. The mitochondria provide energy, fueling processes like cell replication. Appendix cancer can hinder these organelles and their function, resulting in life-threatening situations such as constant vomiting after eating a small amount of food, resulting in no ATP production, which is the main source of energy for your cells.
But we know that cells don't work in isolation. They're always talking to each other and using other signaling molecules called ligands that bind to specific receptors on neighboring cells. These signals can trigger a variety of actions like growth, repair, or self-destruction. In appendix cancer, this communication system is disrupted. The ligand, which is usually a growth factor like epidermal growth factor, EGF, is overproduced, and when they bind to the EGF receptor, it triggers phosphorylation, leading to the activation of the PI3K AKT mTOR pathway, fueling rapid tumor growth. To understand how this happens, let's break down the components of this specific pathway. A signal transduction pathway consists of three parts: reception, transduction, and response. Growth factors like EGF bind to the receptor, which phosphorylates and recruits and activates the PI3K. PI3K converts PIP2 in the cell membrane into PIP3, which is a signaling lipid. PIP3 serves as a docking site for AKT, which is then activated by phosphorylation through another kinase, PDK1. Activated AKT phosphorylates downstream and targets mTOR, which is a central regulatory cell that monitors cell growth. mTOR promotes protein synthesis and cell proliferation while inhibiting cell recycling. As a result, cancer cells grow uncontrollably and invade the surrounding tissues.
But what sparks these changes? External factors like chronic inflammation such as appendicitis, genetic predispositions, exposure to carcinogens in the diet or environment can trigger DNA damage, initiating cancer development. The cell cycle has built-in checkpoints to ensure each phase is completed correctly. For example, the G1 checkpoint checks for DNA damage before replication. The G2 checkpoint ensures all DNA is copied accurately before division, and M checkpoint checks to make sure the sister chromatids are correctly attached to the spindle microtubes. In appendix cancer, mutations in the checkpoint regulators like P53 allow cells to bypass these safeguards, dividing uncontrollably.
When the PI3K AKT mTOR pathway is overactive, cells evade apoptosis. Additionally, when the cell's regulation fails, uncontrolled cell division leads to tumor formation, and mutations in the P53 gene, which normally suppress these tumors, are often implicated in appendix cancer. Without P53 cells with damaged DNA proceed through the cycle, accumulating mutations that promote malignancy. It can be disrupted by mutations in the proteins at the M checkpoint. The lack of proper spindle fiber attachment will lead to irregular chromosomes in each daughter cell, which is a common cause of appendix cancer. So how can we use this information to develop treatments? Well, we can intervene in different parts of this process. For example, EGFR inhibitors or antibodies can block the binding of the EGF to its receptor, stopping the cascade before it even begins. Or molecules like the APR246 restore the P53 function, promoting apoptosis in cancer cells. Agents that enhance the spindle fiber attachments like texanes prevent the irregular splitting of chromosomes by stabilizing the spindle microtubules. Understanding appendix cancer at the cellular level reveals opportunities for targeted therapies, and by addressing disrupted pathways and external triggers, we can work towards creating a cure for appendix cancer. Thank you for listening.